As you settle into this moment, notice the weight of your body against the surface beneath you. Feel the air moving gently through your airways, the subtle pulse of blood through your vessels, the quiet hum of sensation from your skin. Behind all of these experiences, behind every perception and every movement, lies an intricate network of tissue that has been shaping your reality since before your first breath. This network is your nervous system, and tonight we explore its architecture not as a distant subject to master, but as an intimate map of the very structure that makes experience possible. Neuroanatomy is the study of the nervous system's physical organization, the arrangement of neurons into circuits, the assembly of circuits into regions, the connection of regions into networks that span from the tip of your spine to the furthest reaches of your cerebral cortex. It asks where things are, how they are arranged, and why this arrangement matters. Each structure we will encounter has a location, a shape, a set of connections that determine what it does and how it contributes to the whole. Think of the nervous system as a vast city that never sleeps, a metropolis of 86 billion neurons, each one a citizen with connections to thousands of neighbors. This city has districts dedicated to different functions, some processing vision, others coordinating movement, still others storing memories or regulating emotions. It has highways of white matter carrying signals between distant regions, local streets of gray matter where processing occurs, and communication hubs where information from multiple sources converges and decisions are made. The architecture of this city determines the flow of traffic, the patterns of activity, the emergence of everything you think and feel and do. We will also think of the nervous system as a landscape, with mountains and valleys, rivers and plains, each feature shaped by the forces that created it. The folds of the cerebral cortex rise like ridges carved by evolution's patient work. The ventricles flow like underground rivers, carrying cerebrospinal fluid through caverns deep within the brain. The spinal cord extends like a great trunk from which branches emerge, reaching out to every corner of the body. This landscape is not random, but reflects the solutions that nature has found to the problem of coordinating a complex organism. And we will see the nervous system as a communication network, more sophisticated than any human technology, with signals traveling along fibers, jumping across synapses, converging and diverging through circuits of extraordinary complexity. This network processes information from the external world and internal body, generates commands that produce movement, maintains functions necessary for survival, and creates the substrate for consciousness itself. The nervous system divides into two major components based on location. The central nervous system consists of the brain and spinal cord, protected by bone and surrounded by membranes and fluid. The peripheral nervous system includes all neural tissue outside this central core, the nerves that extend throughout the body, connecting the central nervous system to muscles, glands, and sensory receptors. These two components work as an integrated unit, with the peripheral system gathering information and carrying out commands, while the central system processes information and generates responses. Let us begin with the fundamental building block, the neuron itself. Though we are exploring anatomy rather than cellular function, understanding the neuron's structure is essential for understanding how neurons organize into larger systems. A neuron consists of a cell body containing the nucleus and most organelles, dendrites that branch like the crown of a tree to receive signals from other neurons. and an axon that extends away from the cell body to transmit signals to other cells. The axon can be remarkably long. Some axons extend from your lower back all the way to your toes, a distance of more than a meter, making neurons among the longest cells in the body. Axons are often wrapped in myelin, a fatty insulating material that appears white when many myelinated axons are bundled together. This is why we speak of white matter and gray matter in the nervous system. White matter consists primarily of myelinated axons, the communication cables connecting different regions. Gray matter consists primarily of neuron cell bodies, dendrites, and unmyelinated axons, the processing centers where computations occur. Throughout the central nervous system, these two types of tissue are arranged in patterns that reflect function, with gray matter regions processing information and white matter tracts connecting them. Neurons do not exist in isolation, but are supported by glial cells, which outnumber neurons and perform crucial functions. In the central nervous system, astrocytes regulate the chemical environment around neurons. Oligodendrocytes produce the myelin that wraps axons, and microglia serve as immune cells. In the peripheral nervous system, Schwann cells produce myelin. These glial cells were once thought to be merely supportive, but we now recognize that they actively contribute to neural function, influencing signal transmission, synaptic formation, and information processing. The spinal cord serves as the major conduit between the brain and the body, and its anatomy reflects this role. It extends from the base of the skull down through the vertebral column, protected by the bony vertebrae that surround it. In cross-section, the spinal cord shows a characteristic butterfly-shaped region of gray matter surrounded by white matter. The gray matter contains neuron cell bodies, motor neurons that send commands to muscles, interneurons that process information locally, and the terminals of sensory neurons whose cell bodies lie outside the cord. The white matter contains ascending tracts carrying sensory information toward the brain and descending tracts carrying motor commands from the brain. The spinal cord is organized segmentally, with 31 pairs of spinal nerves emerging at regular intervals. Each spinal nerve connects to a specific region of the body, forming a dermatomal map. Each segment of the cord receives sensory input from and sends motor output to a particular strip of skin and underlying muscle. This segmental organization reflects our developmental origins and provides a systematic way for the central nervous system to monitor and control the entire body. Sensory information enters the spinal cord through dorsal roots, where the cell bodies of sensory neurons cluster in dorsal root ganglia. Motor commands exit through ventral roots, carried by axons of motor neurons whose cell bodies reside in the ventral horn of the spinal cord's gray matter. This separation of sensory input and motor output is a fundamental organizing principle, maintained throughout much of the nervous system. The spinal cord does not simply relay information. It also processes certain reflexes locally. When you touch something hot and your hand jerks away before you consciously register pain, this withdrawal reflex is coordinated by circuits within the spinal cord. Sensory neurons detecting the harmful stimulus connect to interneurons in the cord, which activate motor neurons to muscles that pull your hand away. This local processing allows for rapid protective responses without waiting for signals to travel to the brain and back. As we move upward from the spinal cord, we encounter the brainstem, the region that forms the cord's continuation into the skull. The brainstem consists of three sections: the medulla oblongata, the pons, and the midbrain, each with distinct anatomy and functions. Despite being relatively small, the brainstem contains centers that control vital functions, pathways carrying information between the spinal cord and higher brain regions, and nuclei that give rise to most of the cranial nerves. The medulla oblongata is the lowest portion of the brainstem, directly continuous with the spinal cord. Its external surface shows pyramidal ridges formed by descending motor tracts that cross from one side to the other at the pyramidal decussation. This crossing means that the right side of the brain controls the left side of the body and vice versa, a pattern that holds throughout most of the nervous system. Within the medulla lie clusters of neurons that regulate cardiovascular function, breathing, and other autonomic processes. The medulla also contains nuclei that receive input from and send output to structures in the head and neck through cranial nerves. Above the medulla lies the pons, a bulging region whose name means bridge in Latin, reflecting its role in connecting different parts of the brain. The pons contains nuclei involved in regulating breathing, facial sensation and movement, and other functions. It also contains massive bundles of fibers that connect the cerebral cortex to the cerebellum, the structure attached to the back of the brainstem that coordinates movement. The midbrain, the uppermost portion of the brainstem, contains structures involved in visual and auditory processing, motor control, and arousal. Two prominent bumps on its dorsal surface, the superior and inferior colliculi, process visual and auditory information respectively. The midbrain also contains the substantia nigra, a darkly pigmented region whose neurons produce dopamine and project to other brain areas involved in movement control. Damage to these neurons leads to Parkinson's disease, illustrating how anatomical structures and their connections determine function and how disruption of specific regions produces specific deficits. Running through the core of the brainstem is the reticular formation, a diffuse network of neurons with widespread connections throughout the brain. The reticular formation influences arousal and consciousness, filtering sensory information and modulating the brain's overall state of activation. It is not a discrete structure with clear boundaries, but rather a network embedded within the brainstem, illustrating that not all neural organization follows a simple, compartmentalized pattern. The cranial nerves emerge from the brainstem and provide sensory input from and motor output to the head, neck, and some internal organs. There are 12 pairs of cranial nerves, numbered using Roman numerals and often remembered by their functions. The olfactory nerve carries smell information from the nose to the brain. The optic nerve carries visual information from the eyes. The oculomotor, trochlear, and abducens nerves control eye movements. The trigeminal nerve provides sensation from the face and controls the muscles of chewing. The facial nerve controls facial expression and carries taste information from the front of the tongue. The vestibulocochlear nerve carries information about hearing and balance from the inner ear. The glossopharyngeal and vagus nerves have complex functions including taste, swallowing, and autonomic control of internal organs. The accessory nerve controls certain neck and shoulder muscles, and the hypoglossal nerve controls tongue movements. The vagus nerve deserves special mention as it extends far beyond the head, sending branches to the heart, lungs, and digestive organs throughout the abdomen. It is the major component of the parasympathetic division of the autonomic nervous system, slowing heart rate, stimulating digestion, and generally promoting rest and recovery. The vagus nerve illustrates how cranial nerves, despite their name suggesting they serve only the cranium, can have far-reaching effects throughout the body. Attached to the posterior aspect of the brainstem is the cerebellum, a structure whose name means little brain. The cerebellum is aptly named as it resembles a miniature version of the cerebrum with a highly folded outer cortex, internal white matter, and deep nuclei. Despite comprising only about 10% of the brain's volume, the cerebellum contains more than half of the brain's neurons, packed densely in its cortex. The cerebellum's surface is marked by narrow parallel folds called folia, separated by deep fissures. These folds dramatically increase surface area, allowing more cortical tissue to fit within the posterior cranial fossa. The cerebellar cortex has a remarkably regular structure, with the same pattern of cells and connections repeated throughout, suggesting a common computational operation applied to different types of information. The cerebellum receives input from the spinal cord, carrying information about body position and movement, from the cerebral cortex carrying information about intended movements, and from the vestibular system carrying information about balance and head position. It integrates this information and sends output back to motor areas of the cortex and to motor nuclei in the brainstem. Through these connections, the cerebellum coordinates movement, ensuring that actions are smooth, accurate, and appropriately timed. It also contributes to motor learning, the process by which movements become more skilled with practice. Damage to the cerebellum does not cause paralysis, but rather impairs coordination. Movements become jerky and inaccurate, balance is disturbed, and fine motor control is lost. The specific deficits depend on which part of the cerebellum is damaged, as different regions process information from different body parts and contribute to different aspects of motor control. This regional specialization is another recurring principle. Different parts of structures have different connections and therefore different functions. Superior to the brainstem lies the diencephalon, a region largely hidden from external view but playing crucial roles in sensory processing, homeostatic regulation, and consciousness. The diencephalon consists primarily of the thalamus and hypothalamus, two structures with very different functions despite their anatomical proximity. The thalamus consists of two oval masses of gray matter, one on each side of the brain's midline, separated by a fluid-filled space called the third ventricle. The thalamus is sometimes called the brain's relay station because most sensory information ascending to the cerebral cortex passes through thalamic nuclei. Different thalamic nuclei receive different types of sensory input. Visual information goes to the lateral geniculate nucleus, auditory information to the medial geniculate nucleus, touch and other body sensations to the ventral posterior nucleus. From these nuclei, information is relayed to appropriate regions of the cerebral cortex for further processing. But the thalamus does more than simply relay information. It filters, modulates, and integrates it. Thalamic nuclei receive feedback from the cortical areas they project to, creating loops that can enhance certain signals while suppressing others. The thalamus plays a role in directing attention, in regulating consciousness and arousal, and in coordinating activity across cortical regions. Its anatomy, multiple distinct nuclei with specific connections, reflects these diverse functions. The hypothalamus lies below the thalamus, forming the floor and part of the walls of the third ventricle. Despite being small, roughly the size of an almond, The hypothalamus contains numerous nuclei that regulate fundamental aspects of physiology and behavior. It controls body temperature, hunger and thirst, circadian rhythms, stress responses, and reproductive functions. It links the nervous and endocrine systems, producing hormones that control the pituitary gland, which in turn regulates other endocrine glands throughout the body. The hypothalamus receives input about the internal state of the body, blood temperature, osmolarity, glucose levels, hormone concentrations. It also receives input from other brain regions about external conditions and emotional states. Based on this information, it activates appropriate responses through both neural and hormonal pathways. When body temperature rises, the hypothalamus activates mechanisms to promote heat loss. When blood osmolarity increases, it triggers thirst and the release of hormones that conserve water. This integration of sensory input with homeostatic control exemplifies how anatomical connections determine function. The epithalamus, another component of the diencephalon, includes the pineal gland, which produces melatonin and helps regulate circadian rhythms. The subthalamus contains nuclei involved in motor control. These smaller divisions of the diencephalon contribute to the region's functional diversity. Now we turn to the cerebrum, the largest part of the human brain, divided into two hemispheres connected by a massive bundle of white matter fibers called the corpus callosum. The cerebrum's outer surface is the cerebral cortex, a layer of gray matter folded into gyri and sulci, ridges and grooves that increase surface area. If the cortex were unfolded and flattened, it would cover an area of roughly 2,000 to 2,500 square centimeters, about the size of a large pillowcase, all fitted within the skull through this intricate folding. The pattern of gyri and sulci is consistent enough across individuals that major landmarks can be identified and named, allowing neuroanatomists and clinicians to communicate about locations. Certain deep sulci divide each hemisphere into lobes, named after the skull bones that overlie them. The central sulcus runs from the top of the hemisphere down its lateral surface, separating the frontal lobe from the parietal lobe. The lateral sulcus separates the frontal and parietal lobes from the temporal lobe below. The parieto-occipital sulcus, visible on the medial surface of the hemisphere, separates the parietal lobe from the occipital lobe at the back of the brain. These lobes are not arbitrary divisions, but reflect functional organization to some degree. The frontal lobe contains motor areas that control voluntary movement, along with regions involved in planning, decision-making, and personality. The parietal lobe processes sensory information from the body and contributes to spatial awareness. The temporal lobe processes auditory information and plays roles in memory and emotion. The occipital lobe is dedicated to visual processing. However, these are generalizations. Each lobe contains multiple distinct regions with specific functions, and many functions involve cooperation between lobes. Just anterior to the central sulcus lies the precentral gyrus, which contains the primary motor cortex. This region sends commands that initiate voluntary movements. The primary motor cortex is organized somatotopically, meaning different parts of the body are controlled by different regions of the cortex, arranged in an orderly map. Neurons controlling the feet are located near the midline at the top of the hemisphere, while those controlling the face are located more laterally and inferiorly. The amount of cortex devoted to controlling different body parts is not proportional to their size, but to the precision of control required. The hands and face, which perform fine, skilled movements, occupy a disproportionately large cortical area. This distorted representation can be depicted as a homunculus, a little human figure draped over the cortex with body parts sized according to their cortical representation. The motor homunculus has enormous hands and a huge face, tiny trunk and legs, reflecting cortical organization. A similar sensory homunculus exists in the postcentral gyrus, the primary somatosensory cortex that receives sensory information from the body. The somatosensory homunculus is similarly distorted, with large representations for the hands and face, areas where sensory discrimination is finest. The primary motor and somatosensory cortices are surrounded by secondary motor and sensory areas. Premotor cortex, supplementary motor area, and somatosensory association areas. These regions process information at higher levels, planning movements, integrating sensory input from different modalities, and contributing to complex functions like recognizing objects by touch or coordinating sequences of movements. The occipital lobe, at the back of the hemisphere, is devoted primarily to vision. The primary visual cortex, located along and near the calcarine sulcus on the medial surface of the occipital lobe, receives input from the thalamus carrying visual information from the eyes. Like other primary sensory areas, the primary visual cortex is organized topographically, with adjacent regions of visual space represented in adjacent regions of cortex. The center of your visual field is represented in the posterior pole of the occipital lobe, while peripheral vision maps to more anterior regions. Surrounding the primary visual cortex are visual association areas that process different aspects of visual information: color, motion, form, spatial relationships. Some of these areas extend into the parietal and temporal lobes, reflecting the distributed nature of visual processing. Damage to different visual areas produces different deficits: loss of color vision, inability to perceive motion, failure to recognize faces, demonstrating how specific anatomical regions subserve specific functions. The temporal lobe contains the primary auditory cortex on the superior temporal gyrus, receiving auditory information from the thalamus. Like other sensory cortices, it is organized topographically, with different frequencies of sound represented in different locations. The temporal lobe also contains regions crucial for memory formation, particularly the hippocampus and surrounding medial temporal lobe structures, which we will explore in more detail later. The anterior temporal lobe contributes to semantic memory, knowledge of facts and concepts. The inferior temporal cortex participates in object recognition, integrating visual features to identify what we see. The frontal lobe, the largest lobe, contains not only motor areas, but also extensive prefrontal cortex involved in higher cognitive functions. The prefrontal cortex is not a single entity, but includes multiple regions with different connections and functions. The dorsolateral prefrontal cortex contributes to working memory, planning, and cognitive control. The ventromedial prefrontal cortex is involved in decision-making and emotion regulation. The orbitofrontal cortex, on the inferior surface of the frontal lobe, processes reward information and contributes to behavioral flexibility. These prefrontal regions are highly developed in humans and underlie many of our most sophisticated cognitive abilities. The parietal lobe integrates sensory information and contributes to spatial cognition. Beyond the primary somatosensory cortex, the parietal lobe contains regions that integrate touch with vision, create representations of space, and guide attention. The posterior parietal cortex contributes to spatial awareness and the ability to reach toward objects under visual guidance. Damage to parietal regions can produce neglect syndromes where patients fail to attend to one side of space, or apraxias where the ability to perform learned skilled movements is impaired despite intact strength and sensation. An important region at the junction of the parietal, temporal, and occipital lobes is the angular gyrus, involved in reading, mathematical cognition, and other complex functions requiring integration of information from multiple sensory modalities. This region exemplifies how some functions emerge from the interaction of multiple systems rather than being localized to a single anatomical structure. Buried within the lateral sulcus, hidden from surface view, is the insular cortex or insula. To see the insula, one would need to pull apart the frontal and temporal lobes that overlie it. The insula receives input related to taste, visceral sensations from internal organs, and pain. It contributes to emotional awareness, particularly awareness of bodily states, and plays roles in addiction, empathy, and other complex functions. Its hidden location reflects its ancient evolutionary origins and its fundamental role in processing information about the internal state of the body. The cerebral cortex is not a uniform sheet, but contains 6 layers, numbered from the surface inward. Different layers contain different types of neurons with different patterns of connections. Layer 4 primarily receives input from the thalamus. Layers 2 and 3 send connections to other cortical areas in the same or opposite hemisphere. Layers 5 and 6 send connections to subcortical structures like the thalamus, brainstem, and spinal cord. This laminar organization is consistent across much of the cortex, though the thickness and prominence of different layers varies between regions, reflecting differences in function. Regions that receive heavy sensory input have a thick layer 4, while motor regions have a prominent layer 5 from which descending motor commands originate. Beneath the cerebral cortex lies white matter, consisting of axons connecting different cortical regions or connecting cortex to subcortical structures. Association fibers connect different regions within the same hemisphere. Short fibers connecting adjacent gyri, and long fiber bundles connecting more distant regions. Commissural fibers connect the 2 hemispheres, with the corpus callosum being the largest commissure, containing approximately 200 million axons. Projection fibers carry information between cortex and subcortical structures, descending to the thalamus, brainstem, and spinal cord, or ascending from these structures to cortex. Deep within each hemisphere, buried beneath the cortex and white matter, lie clusters of gray matter called the basal ganglia. These include the caudate nucleus and putamen, collectively termed the striatum, the globus pallidus, the subthalamic nucleus, and the substantia nigra. The basal ganglia form loops with the cerebral cortex, receiving input from widespread cortical areas, processing it through their internal circuits, and sending output back to cortex via the thalamus. The basal ganglia are crucial for movement control, particularly for the initiation and scaling of movements and for motor learning. Disorders of the basal ganglia produce characteristic movement abnormalities. Parkinson's disease involves degeneration of dopamine neurons in the substantia nigra and causes difficulty initiating movement, tremor, and rigidity. Huntington's disease involves degeneration of striatal neurons and causes involuntary writhing movements. These clinical syndromes demonstrate the basal ganglia's essential role in normal motor function, but the basal ganglia do more than control movement. Different regions of the basal ganglia connect to different cortical areas and participate in different functions. Connections with prefrontal cortex contribute to cognitive control and decision-making. Connections with limbic regions influence motivation and reward processing. The anatomy of these circuits, which structures connect to which the neurotransmitters they use, how they modulate each other's activity, determines their function and helps explain both normal behavior and the symptoms that arise when these circuits are damaged. Adjacent to the basal ganglia, on the medial surface of the temporal lobe, lies the limbic system, a collection of structures involved in emotion, memory, and motivation. The term limbic comes from the Latin for border, as these structures form a border around the brainstem. The limbic system includes the amygdala, hippocampus, mammillary bodies, cingulate cortex, and other interconnected regions. The amygdala is an almond-shaped structure in the anterior temporal lobe that processes emotional information, particularly fear and threat detection. It receives sensory input from multiple modalities and has connections to widespread brain regions, including the hypothalamus, allowing it to trigger physiological responses to emotional stimuli. The amygdala's connections with the prefrontal cortex and hippocampus allow emotional significance to influence decision-making and memory formation. Emotional events are remembered better than neutral ones, a phenomenon that depends on amygdala-hippocampus interactions. The hippocampus, a seahorse-shaped structure curving through the medial temporal lobe, is essential for forming new memories of facts and events. Information from widespread cortical areas converges on the hippocampus through a pathway involving the entorhinal cortex. The hippocampus processes this information and sends outputs back to cortical areas, facilitating the consolidation of memories that can eventually be stored in cortex independently of the hippocampus. Damage to the hippocampus, as occurred famously in patient HM, whose hippocampi were removed surgically, produces profound anterograde amnesia, the inability to form new long-term memories, while leaving older memories and other cognitive functions relatively intact. The internal structure of the hippocampus reveals elegant organization. It contains regions designated CA1, CA2, and CA3, and the dentate gyrus, arranged in a circuit that processes incoming information through successive stages. This circuit exhibits synaptic plasticity, the ability of synapses to strengthen or weaken based on activity, which is thought to underlie its role in memory formation. The cingulate cortex, a band of cortex above the corpus callosum, encircles the diencephalon and has connections with many limbic structures. It participates in emotional processing, pain perception, and cognitive control. The anterior cingulate is involved in detecting conflict between competing responses and in regulating emotional responses. The posterior cingulate contributes to memory and spatial orientation. These limbic structures are interconnected through complex pathways. The fornix, a C-shaped bundle of fibers, carries output from the hippocampus to the mammillary bodies of the hypothalamus and to other targets. The mammillothalamic tract connects the mammillary bodies to anterior thalamic nuclei, which project to the cingulate cortex, which sends connections back to the hippocampus, forming a circuit known as the Papez circuit that contributes to memory and emotion. Understanding these anatomical connections helps explain how damage to specific regions produces specific deficits. Damage to the hippocampus impairs memory formation. Damage to the fornix, which carries hippocampal output, produces similar deficits. Damage to the mammillary bodies, often seen in chronic alcoholism, also impairs memory. The anatomy reveals the pathways through which information flows, and disruption anywhere along these pathways can impair the functions they support. Throughout this exploration, we see anatomical principles recurring. Structures are organized topographically with orderly maps of sensory surfaces or motor outputs. Different regions are specialized for different functions, with specialization reflected in their connections. Information flows through pathways that connect structures in specific sequences. Feedback connections create loops that modulate processing. And the same basic elements, neurons, synapses, circuits, are organized in different patterns to create different computational capabilities. The neuroanatomical landscape we are mapping is not arbitrary, but reflects both evolutionary history and functional requirements. Structures that emerged earlier in evolution tend to lie deeper and more medially, while newer structures lie more superficially and laterally. The brainstem and limbic system are ancient, present in some form in all vertebrates. The cerebral cortex is newer, highly expanded in mammals and especially in primates. The prefrontal cortex is most developed in humans, underlying our sophisticated cognitive abilities. As we complete this first portion of our journey through neuroanatomy, we have established foundations, the basic divisions of the nervous system, the structures of the spinal cord and brainstem, the thalamus and hypothalamus, the organization of the cerebral cortex into lobes, the basal ganglia and limbic system. We have begun to see how anatomy determines function, how connections define what structures do, how damage reveals normal roles. The landscape continues to unfold before us, with more regions to explore, more pathways to trace, more principles to discover. But already a picture emerges, the nervous system as an organized whole, with each part connected to others through specific pathways, with information flowing through circuits that transform sensory input into perception, perception into decision, decision into action. The architecture we are mapping is the physical substrate of the mind, the structure that makes thought, feeling, and behavior possible. As you rest in this moment, that structure continues its work, processing the sensory information arising from your body, maintaining the background activities of consciousness, holding the knowledge we are exploring in patterns of neural activity that will, through consolidation, become lasting memories. The neuroanatomy we study is not distant or abstract, but intimately present, the very tissue that enables you to understand these words, to form thoughts about them, to exist as a conscious being embedded in the world. We have traced the fundamental divisions of the nervous system, observed the organization of the spinal cord and brainstem, explored the cerebral cortex's lobes and the deep structures buried beneath them. Now we continue by examining the pathways that carry information through this landscape, the sensory systems that bring the world into the brain, and the motor systems that allow the brain to act upon the world. Information reaches the central nervous system through multiple sensory modalities, each with its own specialized receptors, pathways, and processing centers. Despite their differences, sensory systems share common organizational principles. Receptors transduce physical or chemical stimuli into neural signals. These signals travel along specific pathways to the central nervous system. Information is processed through a hierarchy of structures, with each level extracting different features or performing different computations. and most sensory information passes through the thalamus before reaching the cerebral cortex, where conscious perception arises. Consider the pathway that carries information about touch, pressure, vibration, and proprioception, the sense of body position. Receptors in the skin, muscles, and joints detect these stimuli and send signals along axons whose cell bodies reside in dorsal root ganglia. These axons enter the spinal cord through dorsal roots, but rather than synapsing immediately, they ascend through the spinal cord in white matter tracts called the dorsal columns. The dorsal columns are organized topographically with information from lower body parts traveling in the medial portion called the gracile fasciculus, while information from upper body parts travels in the lateral portion called the cuneate fasciculus. These ascending axons travel all the way to the medulla, where they synapse in the gracile and cuneate nuclei. From these nuclei, second-order neurons send their axons across the midline to the opposite side of the brainstem, forming a crossing called the medial lemniscus. The medial lemniscus ascends through the brainstem to the ventral posterior nucleus of the thalamus. From there, third-order neurons project through white matter to the primary somatosensory cortex in the postcentral gyrus. This three-neuron pathway illustrates several principles. First, the crossing of information means that each side of the brain receives input from the opposite side of the body. Second, the pathway is topographically organized at every level. The orderly arrangement of body parts is maintained in the dorsal columns, in the thalamic nucleus, and in the cortical representation. Third, processing occurs at multiple stages. In the brainstem nuclei, where some filtering and modulation occurs, in the thalamus, which gates information flow to cortex, and in cortex, where complex processing creates the rich perceptual experience of touch. A parallel pathway carries information about pain and temperature. Receptors sensitive to tissue damage and to heat or cold send signals along small diameter axons to the spinal cord. Unlike touch information, these axons synapse immediately upon entering the cord, in the dorsal horn where interneurons process the signals. Second-order neurons then send their axons across to the opposite side of the cord and ascend in the anterolateral portion of white matter, forming the spinothalamic tract. This tract ascends through the brainstem to the thalamus, where third-order neurons project to the somatosensory cortex and to other cortical areas involved in the emotional aspects of pain. The existence of separate pathways for different types of somatic sensation explains certain clinical phenomena. Damage to the dorsal columns impairs fine touch and proprioception on the same side of the body below the lesion, while pain and temperature sensation remain intact. Damage to the anterolateral cord impairs pain and temperature sensation on the opposite side while preserving fine touch. These dissociations reveal the anatomical separation of pathways carrying different information. The visual system demonstrates another variation on the theme of hierarchical, topographic organization. Light striking the retina activates photoreceptors, rods and cones, which convert light into neural signals. These signals pass through several layers of retinal neurons before reaching retinal ganglion cells, whose axons form the optic nerve. The optic nerves from the two eyes converge at the optic chiasm, where axons from the nasal half of each retina cross to the opposite side, while axons from the temporal half remain on the same side. This partial crossing means that each hemisphere receives information from the opposite visual field. the left hemisphere receives input from the right visual field of both eyes, and vice versa. After the chiasm, the fibers form the optic tracts, which project to the lateral geniculate nucleus of the thalamus. From there, neurons send axons through a large white matter bundle called the optic radiation to the primary visual cortex in the occipital lobe. Throughout this pathway, retinotopic organization is preserved. Adjacent points in visual space are represented in adjacent neurons, creating maps of the visual field at each processing stage. The primary visual cortex is not the end of visual processing, but the beginning of cortical analysis. Information flows from primary visual cortex to surrounding visual association areas, which are organized into two broad streams. The dorsal stream projects toward parietal cortex and processes information about where objects are located and how to interact with them, the where or how pathway. The ventral stream projects toward temporal cortex and processes information about what objects are, identifying and recognizing them, the what pathway. This division of labor allows specialized processing of different aspects of visual information. Some retinal ganglion cell axons project not to the lateral geniculate nucleus, but to the superior colliculus in the midbrain. The superior colliculus creates a map of visual space and coordinates eye movements toward objects of interest. It also receives input from other sensory modalities, creating multisensory maps used to orient attention and guide movements. This pathway operates largely below conscious awareness, enabling reflexive eye movements and spatial orienting. The auditory system begins with the cochlea, a spiral structure in the inner ear where sound vibrations are converted into neural signals. Hair cells in the cochlea are arranged tonotopically, with different positions along the spiral responding to different frequencies of sound. This tonotopic organization is preserved throughout the auditory pathway. Axons from the cochlear nerve enter the brainstem and synapse in the cochlear nuclei. From there, information follows a complex pathway with multiple synaptic stations in the brainstem before reaching the inferior colliculus in the midbrain. Then the medial geniculate nucleus of the thalamus, and finally the primary auditory cortex in the superior temporal gyrus. Unlike most sensory pathways, the auditory system has bilateral projections. Each ear sends information to both hemispheres, though the contralateral projection is stronger. This bilateral representation is important for sound localization, which depends on comparing the timing and intensity of sounds reaching the two ears. Brainstem nuclei perform these comparisons, detecting microsecond differences in arrival time that indicate sound direction. The olfactory system follows a different anatomical plan, reflecting its ancient evolutionary origins. Olfactory receptor neurons in the nasal epithelium send axons directly to the olfactory bulb, a structure on the inferior surface of the frontal lobe. The olfactory bulb performs initial processing, then sends projections to several cortical and subcortical areas, including the piriform cortex, amygdala, and entorhinal cortex. Notably, olfactory information reaches cortex without passing through the thalamus, unlike other sensory modalities. The olfactory system's direct connections to the amygdala and hippocampus may explain why smells can evoke powerful emotional memories. Taste information follows yet another path. Taste receptors on the tongue send signals via cranial nerves to the gustatory nucleus in the medulla. From there, information projects to the ventral posterior medial nucleus of the thalamus and then to the gustatory cortex in the insula and frontal operculum. taste, and smell interact extensively, with much of what we perceive as flavor arising from olfactory input rather than taste alone. The vestibular system, which detects head position and movement, shares the 8th cranial nerve with the auditory system. Hair cells in the semicircular canals and otolith organs of the inner ear detect rotational and linear accelerations. This information projects to vestibular nuclei in the brainstem, which have extensive connections to the cerebellum, spinal cord, and motor nuclei controlling eye movements. The vestibular system operates largely unconsciously, maintaining balance and stabilizing gaze during head movements. Some vestibular information reaches the cortex, contributing to spatial orientation and the sense of self-motion. Having explored sensory pathways that bring information into the nervous system, we turn now to motor pathways that carry commands outward to muscles. The primary motor cortex in the precentral gyrus generates the neural commands that initiate voluntary movements. Neurons in layer 5 of the motor cortex send axons down through white matter, forming a massive bundle called the corticospinal tract. The corticospinal tract descends through the internal capsule, a white matter region between the thalamus and basal ganglia, where descending and ascending fibers are tightly packed. It continues through the cerebral peduncles in the midbrain, the basilar pons, where it appears fragmented as fibers weave between pontine nuclei, and into the medulla, where it forms the pyramids on the ventral surface. At the junction between medulla and spinal cord, most corticospinal fibers cross to the opposite side at the pyramidal decussation. The crossed fibers descend in the lateral portion of the spinal cord's white matter, forming the lateral corticospinal tract. The small percentage of fibers that do not cross descend in the anterior corticospinal tract. Corticospinal fibers synapse on interneurons and motor neurons in the spinal cord's ventral horn. These spinal motor neurons send axons out through ventral roots to skeletal muscles, where they form neuromuscular junctions and trigger muscle contractions. This pathway from cortex to muscle allows voluntary control of movement, with damage to the corticospinal tract causing weakness or paralysis on the opposite side of the body. But the corticospinal tract is not the only motor pathway. The corticobulbar tract carries similar commands from motor cortex to motor nuclei of cranial nerves in the brainstem, controlling muscles of the face, jaw, tongue, and larynx. Other descending pathways originating in the brainstem carry commands that control posture and gross movements. The vestibulospinal tract carries signals from vestibular nuclei to spinal motor neurons, adjusting muscle tone to maintain balance. The reticulospinal tract carries signals from the reticular formation, modulating movement and posture based on the overall state of arousal and intention. These multiple descending pathways interact with local spinal circuits that generate basic motor patterns. The spinal cord is not merely a passive relay, but contains circuits that can produce rhythmic movements like walking. Descending commands from the brain initiate, modulate, and guide these spinal circuits rather than specifying every detail of muscle activation. The cerebellum plays a crucial role in motor control, despite not directly sending commands to muscles. It receives copies of motor commands from the cortex, sensory feedback about actual movements from the spinal cord, and vestibular information about balance. By comparing intended movements with actual movements, the cerebellum detects and corrects errors, ensuring that movements are smooth and accurate. Cerebellar output projects back to motor cortex via the thalamus and to brainstem motor nuclei, modulating ongoing movements. The cerebellum is organized into functional regions. The vestibulocerebellum, comprising the flocculonodular lobe, connects with the vestibular system and controls balance and eye movements. The spinocerebellum, including the vermis and intermediate hemisphere, receives input from the spinal cord and regulates limb movements and posture. The cerebrocerebellum, comprising the lateral hemispheres, receives input from cerebral cortex and contributes to motor planning and cognitive functions. This functional specialization reflects different anatomical connections. Different cerebellar regions receive input from and send output to different structures. The basal ganglia, as mentioned earlier, also contribute to motor control through loops with the cortex. The direct pathway through the basal ganglia facilitates intended movements by disinhibiting thalamic neurons that excite motor cortex. The indirect pathway suppresses unwanted movements by increasing inhibition of the thalamus. The balance between these pathways allows for smooth initiation and execution of movements while preventing extraneous movements. Disruption of this balance, as occurs in Parkinson's and Huntington's diseases, produces characteristic movement disorders. Motor control thus emerges not from a single structure but from the interaction of multiple systems, cortex generating commands, basal ganglia selecting and initiating actions, cerebellum coordinating and refining movements, brainstem and spinal circuits executing patterns and maintaining posture. The anatomical connections between these structures determine how they interact and how motor control emerges from their collective activity. Beyond sensory and motor systems, the nervous system contains pathways that regulate autonomic functions, the involuntary processes that maintain homeostasis. The autonomic nervous system consists of two divisions with opposing effects. The sympathetic division, which prepares the body for activity and stress, and the parasympathetic division, which promotes rest and recovery. The sympathetic division originates from the thoracic and lumbar regions of the spinal cord. Preganglionic neurons in the lateral horn of the spinal cord send axons out through ventral roots to ganglia of the sympathetic chain, which runs alongside the vertebral column. Some preganglionic fibers synapse in sympathetic chain ganglia, while others pass through and synapse in more distant collateral ganglia. Postganglionic neurons then send axons to target organs: the heart, blood vessels, lungs, digestive organs, sweat glands, and others. Activation of the sympathetic division produces a coordinated response. Heart rate and contractility increase, airways dilate, blood vessels in muscles dilate while those in digestive organs constrict. The liver releases glucose, sweat glands activate, pupils dilate. These changes prepare the body for physical or mental exertion, supporting the fight or flight response. The anatomical arrangement, with preganglionic neurons clustered in a specific spinal region and connecting to a chain of ganglia, allows for widespread, coordinated activation. The parasympathetic division originates from the brainstem and sacral spinal cord. Cranial nerves, particularly the vagus nerve, carry preganglionic parasympathetic fibers to ganglia near or within target organs in the head, neck, chest, and abdomen. Sacral preganglionic neurons send fibers to pelvic organs. Because parasympathetic ganglia are located close to target organs, postganglionic fibers are short. Parasympathetic activation slows heart rate, constricts airways, stimulates digestion, promotes elimination, and conserves energy. Most organs receive both sympathetic and parasympathetic innervation, allowing fine control through reciprocal regulation. The heart receives sympathetic input that increases rate and parasympathetic input that decreases it, with the actual rate reflecting the balance between these influences. The anatomical pathways, different origins in the central nervous system, different ganglia, different neurotransmitters at target organs, allow these two divisions to produce opposite effects on the same organs. The hypothalamus coordinates autonomic function, receiving information about internal state and external conditions, and activating appropriate autonomic responses through descending pathways to brainstem and spinal autonomic centers. This integration of autonomic control with other homeostatic mechanisms reflects the hypothalamus's role as a master regulator. The organization of white matter pathways throughout the brain reveals the extensive connectivity that allows information to flow between distant regions. Association fibers connect different areas within the same hemisphere. Short association fibers, or U-fibers, connect adjacent gyri, arching through the white matter beneath intervening sulci. Longer association bundles connect more distant regions. The superior longitudinal fasciculus connects frontal and parietal lobes with temporal and occipital lobes, carrying information involved in language, spatial attention, and other functions. The arcuate fasciculus, part of this system, connects language areas in the frontal and temporal lobes. The inferior longitudinal fasciculus connects occipital and temporal lobes, carrying visual information to areas involved in object recognition. The uncinate fasciculus connects frontal and temporal lobes, contributing to emotional processing and memory. Commissural fibers connect the two hemispheres, allowing them to communicate and coordinate their activities. The corpus callosum is by far the largest commissure, containing axons that connect corresponding areas of the two hemispheres. The corpus callosum is divided into regions based on which cortical areas they connect. The genu carries fibers connecting frontal lobes. The body connects parietal and temporal regions, and the splenium connects occipital lobes. Other smaller commissures include the anterior commissure, which connects temporal lobes, and the hippocampal commissure, connecting the two hippocampi. The importance of the corpus callosum is dramatically demonstrated in split brain patients who have had this structure severed to treat severe epilepsy. After surgery, the two hemispheres can no longer share information directly. When such patients are shown an image in their left visual field, which projects to the right hemisphere, they cannot verbally name what they saw because language areas are typically in the left hemisphere and the hemispheres cannot communicate. However, they can point to the object with their left hand, which is controlled by the right hemisphere that saw the image. These dissociations reveal how much of normal function depends on interhemispheric communication through the corpus callosum. Projection fibers carry information between cortex and subcortical structures. Ascending projections from the thalamus carry sensory information and other signals to cortex. Descending projections carry motor commands to the brainstem and spinal cord, as well as feedback signals to the thalamus and other structures. These projection fibers pass through the internal capsule, making this a critical region where damage can have devastating effects. A stroke affecting the internal capsule can disrupt both sensory and motor pathways, causing numbness and paralysis. The internal capsule consists of anterior and posterior limbs that meet at an angle called the genu. Different fibers pass through different regions, corticospinal fibers pass through the posterior limb, corticobulbar fibers through the genu, and connections between thalamus and frontal cortex through the anterior limb. This organization means that small lesions in specific locations produce specific deficits, allowing clinicians to localize damage based on symptoms. Throughout the brain, the arrangement of white and gray matter reflects functional organization. In the spinal cord and brainstem, gray matter lies centrally surrounded by white matter. In the cerebrum and cerebellum, gray matter forms the outer cortex with white matter internal. Deep within the cerebrum lie additional gray matter structures: basal ganglia, thalamus, amygdala, hippocampus. This arrangement places processing centers in the cortex and deep nuclei, connected by white matter pathways that allow communication. The ventricles form a system of interconnected, fluid-filled cavities within the brain and spinal cord. There are 4 ventricles, 2 lateral ventricles within the cerebral hemispheres, the 3rd ventricle between the 2 halves of the thalamus, and the 4th ventricle between the pons and cerebellum. The lateral ventricles connect to the 3rd ventricle through interventricular foramina. The 3rd ventricle connects to the 4th through the cerebral aqueduct running through the midbrain. The 4th ventricle opens to the subarachnoid space surrounding the brain through apertures in its roof. The ventricles are lined with ependymal cells and contain choroid plexus, specialized tissue that produces cerebrospinal fluid. This clear, colorless fluid cushions the brain and spinal cord, protecting them from mechanical trauma. It also provides a stable chemical environment and removes metabolic wastes. Cerebrospinal fluid circulates through the ventricular system and subarachnoid space, eventually being absorbed into the venous blood through arachnoid granulations. The brain and spinal cord are surrounded by 3 membranes called meninges. The outermost is the dura mater, a thick, tough layer that adheres to the inside of the skull and forms a protective covering. The middle layer is the arachnoid mater, which loosely covers the brain with web-like extensions. The innermost is the pia mater, a delicate membrane that closely follows the contours of the brain surface, dipping into every sulcus. Between the arachnoid and pia is the subarachnoid space, filled with cerebrospinal fluid. Vessels supplying the brain travel through this space before penetrating the brain tissue. Blood supply to the brain comes from 2 main sources. The internal carotid arteries ascend through the neck and enter the skull, supplying the anterior and much of the middle portions of the cerebral hemispheres. The vertebral arteries ascend through the cervical vertebrae, enter the skull, and merge to form the basilar artery, which supplies the brainstem and cerebellum before dividing into posterior cerebral arteries that supply the posterior and inferior portions of the hemispheres. These arteries connect at the base of the brain through a ring of vessels called the circle of Willis. This circular anastomosis allows blood to flow from one side to the other and from anterior to posterior circulation, providing collateral circulation if one artery becomes blocked. Despite this protective arrangement, strokes remain common, occurring when blood supply to brain regions is interrupted, causing tissue damage and loss of function. Different arteries supply different brain regions, and the anatomy of vascular territories explains the patterns of deficits seen in strokes. The anterior cerebral artery supplies medial portions of the frontal and parietal lobes, including leg and foot areas of the motor and sensory cortices. Occlusion of this artery causes weakness and sensory loss, primarily affecting the leg on the opposite side. The middle cerebral artery supplies lateral portions of the hemispheres, including face and arm areas of sensor-motor cortex and much of the temporal lobe. Middle cerebral artery stroke causes face and arm weakness, sensory loss, and often language deficits if the left hemisphere is affected. The posterior cerebral artery supplies the occipital lobe and inferior temporal regions. Its occlusion can cause visual field defects and memory impairment if medial temporal structures are affected. The brain's high metabolic rate demands abundant blood flow. Though comprising only about 2% of body weight, the brain receives approximately 15% of cardiac output and consumes 20% of the body's oxygen. This intense metabolic demand makes the brain especially vulnerable to disruptions in blood supply. Even brief interruptions can cause cellular damage and functional loss. The blood-brain barrier protects the brain's chemical environment. Capillaries in the brain have specialized endothelial cells with tight junctions that prevent most substances from freely crossing from blood into brain tissue. Only small, lipid-soluble molecules like oxygen and carbon dioxide can cross easily. Glucose, the brain's primary fuel, is transported across by specific carriers. This selective barrier protects the brain from toxins and maintains stable conditions necessary for neural function, but it also complicates treatment of brain diseases by preventing many drugs from reaching brain tissue. As this exploration of pathways and systems continues, we see how the nervous system's anatomy enables its functions. Specific receptors detect specific stimuli. Dedicated pathways carry different types of information. Processing occurs through hierarchies of structures, each extracting features or performing computations. Motor commands descend through specific tracts to specific targets. Autonomic pathways regulate visceral functions. White matter connections allow distant regions to communicate. Blood vessels provide the metabolic support neural tissue requires. And protective structures, skull, meninges, cerebrospinal fluid, blood-brain barrier, safeguard this delicate system. The anatomical organization we are mapping reflects both functional requirements and evolutionary history. Pathways are organized to minimize conduction time and maximize efficiency. Processing hierarchies allow for increasingly complex analysis of information. Parallel pathways allow simultaneous processing of different features. Feedback connections allow higher centers to modulate lower ones. Redundancy provides resilience when damage occurs. And the elaboration of structures like the cerebral cortex and cerebellar hemispheres in mammals, and especially in primates and humans, reflects the evolution of increasingly sophisticated cognitive and motor capabilities. Understanding neuroanatomy thus means understanding the physical substrate of behavior and mind. The circuits we trace, the connections we map, the regions we identify. These are not abstract entities but actual tissue whose properties determine what we can perceive, think, and do. Damage to specific regions produces specific deficits, revealing normal function. Imaging studies show which regions activate during different tasks, revealing functional specialization. And the overall architecture, the pathways, the hierarchies, the networks, constrains and enables the information processing that creates experience and generates behavior. As we continue our journey, more systems and regions await exploration, but already the landscape takes shape. a vast, interconnected network with specialized regions processing different types of information, with pathways carrying signals between structures, with hierarchical organization allowing increasingly complex functions to emerge. This is the architecture of the nervous system, the anatomy that makes the mind possible. We have traversed the major divisions of the nervous system, followed sensory pathways as they carry information inward and motor pathways as they send commands outward, observed the white matter connections linking distant regions. Now we move deeper into the landscape, exploring specialized systems in greater detail and observing how anatomical structures integrate to create complex functions. The language networks of the human brain provide a compelling example of how anatomy determines capability. Language function is typically lateralized, meaning it depends primarily on one hemisphere, usually the left, even in many left-handed individuals. Two cortical regions are particularly crucial. Broca's area, located in the inferior frontal gyrus of the frontal lobe, is involved in language production and grammatical processing. Wernicke's area, located at the junction of the temporal and parietal lobes, is involved in language comprehension. These regions are connected by the arcuate fasciculus, a white matter bundle that allows them to communicate. Damage to Broca's area produces expressive aphasia, where speech production is labored and grammatically impoverished, though comprehension remains relatively intact. Patients with this condition understand what is said to them and know what they want to say, but cannot fluently produce speech. Damage to Wernicke's area produces receptive aphasia, where speech is fluent but meaningless, and comprehension is severely impaired. Patients with this condition produce streams of grammatical sentences that convey no coherent meaning, and they cannot understand spoken or written language. The existence of these distinct syndromes reveals anatomical specialization. Different brain regions support different aspects of language. But language is not localized solely to Broca's and Wernicke's areas. Reading involves visual cortex to recognize written symbols, along with regions that connect visual forms to sounds and meanings. Speaking involves motor cortex and associated regions that control the muscles of articulation. Listening involves auditory cortex. Semantic knowledge, the meanings of words and concepts, involves distributed networks across temporal cortex. Language emerges from the coordinated activity of multiple regions connected through white matter pathways. The arcuate fasciculus connecting frontal and temporal language areas illustrates how white matter pathways are essential for integrated function. Damage to this pathway produces conduction aphasia, where comprehension and spontaneous speech are relatively preserved, but the ability to repeat words or phrases is impaired. This specific deficit reveals the importance of the anatomical connection between regions involved in comprehension and production. Memory systems provide another window into how anatomy underlies function. We have encountered the hippocampus and its role in forming new declarative memories, memories of facts and events that can be consciously recalled. The medial temporal lobe memory system includes not only the hippocampus, but also the surrounding entorhinal, perirenal, and parahippocampal cortices. Information from widespread cortical areas converges on this system through a series of connections. Cortical association areas process sensory information at high levels, extracting features and recognizing patterns. This processed information projects to the perirenal and parahippocampal cortices, which in turn project to the entorhinal cortex. The entorhinal cortex serves as the main gateway to the hippocampus, sending information through a pathway that sequentially engages different hippocampal regions: dentate gyrus, CA3, CA1. From CA1, information flows back to entorhinal cortex and then back to the cortical areas that originally sent the information. This loop allows the hippocampus to bind together different aspects of an experience: what happened, where it happened, what it meant, creating coherent episodic memories. The hippocampus does not permanently store memories, but facilitates their consolidation in cortex. Gradually, over time, cortical connections strengthen and memories can be retrieved without hippocampal involvement. This process explains why hippocampal damage impairs formation of new memories, but largely spares older memories that have already been consolidated. The anatomical separation between the temporary storage system in medial temporal lobe and the distributed long-term storage in cortex creates this temporal gradient. Other memory systems involve different anatomical substrates. Procedural memory, memory for skills and habits, depends on the basal ganglia and cerebellum rather than the hippocampus. Learning to ride a bicycle, play a musical instrument, or type on a keyboard involves these structures. The basal ganglia contribute to learning action sequences and habit formation through their loops with motor and prefrontal cortex. The cerebellum contributes to motor learning and timing. These forms of memory can be intact even when hippocampal damage prevents formation of new declarative memories, demonstrating anatomical dissociation between memory systems. Working memory, the ability to temporarily hold and manipulate information, depends on prefrontal cortex. Different prefrontal regions contribute to different aspects of working memory. Dorsolateral prefrontal cortex maintains information in active states and manipulates it to solve problems. Ventrolateral prefrontal cortex encodes and retrieves information. These regions connect extensively with posterior cortical areas that represent different types of information: visual, auditory, spatial, allowing prefrontal cortex to hold and work with diverse content. Emotional processing and regulation involve networks centered on the amygdala and prefrontal cortex. The amygdala receives sensory information through multiple pathways, including a direct route from sensory thalamus that provides rapid but crude information, and cortical routes that provide slower but more detailed analysis. This dual pathway arrangement allows the amygdala to initiate rapid emotional responses to potential threats, while more detailed processing continues. The amygdala's outputs to hypothalamus, brainstem, and other structures trigger physiological responses: increased heart rate, stress hormone release, heightened attention, that prepare the organism to deal with emotionally significant situations. The prefrontal cortex, particularly ventromedial and orbitofrontal regions, regulates emotional responses through connections with the amygdala. These prefrontal areas can enhance or suppress amygdala activity based on context, goals, and learned associations. Damage to ventromedial prefrontal cortex impairs emotional regulation and decision-making, producing impulsive behavior and poor social judgment despite intact intellectual abilities. The famous case of Phineas Gage, who sustained frontal lobe damage in a 19th century accident and subsequently showed dramatic personality changes, illustrated the role of frontal regions in emotional and social behavior. The anterior cingulate cortex, positioned between prefrontal cortex and limbic structures, integrates emotional and cognitive processing. It activates when conflicts arise between competing responses, signaling the need for increased cognitive control. It also processes the emotional aspects of pain and contributes to error monitoring. The anterior cingulate's anatomical position and connections make it well suited for integrating motivation, emotion, and cognitive control. Attention involves distributed networks that select relevant information from the vast sensory input constantly reaching the brain. The posterior parietal cortex, particularly the intraparietal sulcus region, creates priority maps that represent the locations of salient stimuli. The frontal eye fields in frontal cortex control where attention is directed and generate eye movements toward attended locations. The temporoparietal junction detects unexpected or behaviorally relevant stimuli, triggering attention shifts. These cortical areas interact with subcortical structures, including the pulvinar nucleus of the thalamus and the superior colliculus, to implement attention. The right hemisphere plays a special role in spatial attention. Damage to right parietal cortex often produces hemispatial neglect, where patients fail to attend to or even acknowledge the left side of space. They may eat food only from the right side of a plate, shave only the right side of the face, or draw only the right half of objects. This deficit is not simply sensory loss. They can perceive left-sided stimuli when attention is explicitly directed there, but rather a failure of attention. The anatomical lateralization of spatial attention to the right hemisphere contrasts with the left lateralization of language, showing how different functions can be asymmetrically distributed. Sleep and wakefulness are regulated by networks distributed through the brainstem, hypothalamus, and basal forebrain. The reticular activating system, a network extending through the brainstem, promotes wakefulness through widespread projections to the thalamus and cortex. Different neurotransmitter systems contribute: cholinergic neurons in the brainstem and basal forebrain, noradrenergic neurons in the locus coeruleus, serotonergic neurons in the raphe nuclei, histaminergic neurons in the hypothalamus. These arousal systems are active during waking, maintaining cortical activation and conscious awareness. The hypothalamus contains regions that promote wakefulness and others that promote rest. The lateral hypothalamus contains neurons that produce orexin, a neuropeptide that stabilizes the waking state. Loss of orexin neurons causes narcolepsy, a disorder characterized by sudden transitions between wakefulness and states resembling rapid eye movement patterns. The ventrolateral preoptic nucleus contains neurons that promote restorative states by inhibiting arousal systems. These hypothalamic regions interact with brainstem arousal systems and with the circadian clock in the suprachiasmatic nucleus to regulate the daily cycle of activity and rest. The suprachiasmatic nucleus, a small region above the optic chiasm, serves as the master circadian clock, generating approximately 24-hour rhythms that persist even without external time cues. It receives direct input from the retina through the retinohypothalamic tract, allowing light to synchronize the internal clock to the external day-night cycle. The suprachiasmatic nucleus sends projections to other hypothalamic regions and to the pineal gland, which produces melatonin at night. Through these outputs, the suprachiasmatic nucleus coordinates circadian rhythms in physiology and behavior throughout the body. Pain processing involves multiple pathways and regions, reflecting the complexity of pain as a multidimensional experience. The lateral pain pathway, running through the lateral thalamus to primary somatosensory cortex, processes the sensory discriminative aspects of pain, where it is, how intense, what type. The medial pain pathway, projecting through medial thalamus to anterior cingulate cortex, insula, and other regions, processes the affective motivational aspects. How unpleasant it is, how much it matters, what it means. This anatomical separation explains why pain has both sensory and emotional components, and why these components can be dissociated. Certain interventions can reduce the unpleasantness of pain without reducing its intensity by preferentially affecting the medial pathway. The periaqueductal gray, a region surrounding the cerebral aqueduct in the midbrain, is central to pain modulation. It receives input from cortex and hypothalamus and sends descending projections to the brainstem and spinal cord that can suppress pain signals. This descending pain modulation system uses endogenous opioids and other neurotransmitters to reduce pain transmission. The system can be activated by stress, attention, expectation, and other factors, explaining phenomena like stress-induced analgesia and placebo effects. The anatomy of this system, its connections from higher centers, its descending projections to pain pathways, allows cognitive and emotional factors to influence pain perception. Reward processing and motivation depend on circuits involving the ventral tegmental area, nucleus accumbens, and prefrontal cortex. The ventral tegmental area, located in the midbrain, contains dopamine neurons that project to the nucleus accumbens in the ventral striatum and to prefrontal cortex. These projections signal reward prediction errors. When outcomes are better than expected, dopamine neuron activity increases. When worse than expected, it decreases. Through these signals, the dopamine system supports learning about which actions lead to rewards and which to avoid. The nucleus accumbens integrates dopamine signals with input from the amygdala carrying emotional information and from hippocampus carrying contextual information. It projects to motor regions, influencing which behaviors are expressed based on their expected value. The prefrontal cortex represents goals and strategies, sending signals back to the nucleus accumbens and other structures to guide behavior toward long-term objectives, even when immediate rewards are absent. This circuit anatomy creates a system that can learn from experience, assign value to options, and select actions to achieve goals. Addiction involves pathological changes in this reward circuit. Drugs of abuse directly or indirectly increase dopamine signaling in the nucleus accumbens, creating powerful reinforcement. With repeated exposure, the circuit undergoes long-lasting changes, alterations in synaptic strength, changes in gene expression, structural modifications. These changes make drug-associated cues extremely salient, impair control over drug seeking, and reduce sensitivity to natural rewards. The neuroanatomy of addiction, which circuits are affected, how they change, helps explain why addiction is so difficult to overcome and informs therapeutic approaches. Social behavior depends on networks involving the amygdala, prefrontal cortex, and temporal lobe regions. The superior temporal sulcus processes biological motion, the movements of living things, and is particularly responsive to faces and eye gaze. The fusiform face area in the inferior temporal lobe specializes in face recognition. The amygdala processes the emotional significance of social signals. The medial prefrontal cortex and temporoparietal junction contribute to theory of mind. The ability to infer others' mental states, beliefs, and intentions. These regions activate when people think about what others might be thinking or feeling. The mirror neuron system, discovered in premotor cortex and parietal regions, contains neurons that activate both when performing an action and when observing someone else perform that action. This system may contribute to action understanding, imitation learning, and empathy, though its exact role in humans remains debated. The anatomical distribution of mirror-like responses across motor and sensory regions suggests a network for mapping observed actions onto the observer's own motor system. The development of the nervous system involves a precisely orchestrated sequence of events that creates the adult anatomy. The neural tube, formed early in embryonic development, gives rise to the entire central nervous system. The anterior portion of the neural tube develops three primary vesicles: forebrain, midbrain, and hindbrain. These subdivide further, with the forebrain developing into the telencephalon, which becomes the cerebral hemispheres, and the diencephalon, which becomes the thalamus and hypothalamus. The midbrain remains relatively unchanged. The hindbrain develops into the metencephalon, which becomes the pons and cerebellum, and the myelencephalon, which becomes the medulla. As these regions develop, neurons are born through cell division in zones near the ventricles. These newborn neurons migrate outward to their final positions, guided by radial glial cells and chemical signals. In the cortex, neurons born later migrate past earlier born neurons, creating an inside-out pattern where deeper layers form first and superficial layers last. Once in position, neurons extend axons toward their target regions, navigating through the developing brain using molecular guidance cues. Growth cones at axon tips detect attractive and repulsive signals that steer them along appropriate paths. When axons reach their target regions, they form synapses, establishing the connections that will carry information. Initially, far more synapses form than will ultimately remain. Experience and neural activity determine which synapses strengthen and survive, and which weaken and are eliminated, a process called synaptic pruning. This activity-dependent refinement sculpts circuits to match the environment and the organism's needs. The anatomical connectivity of the adult brain thus reflects both genetic programs that establish the basic pattern and experience-dependent processes that fine-tune connections. The human brain continues developing long after birth. The cerebral cortex increases in volume through childhood and adolescence, with different regions maturing at different rates. Sensory and motor areas mature earliest. Association areas in parietal and temporal cortex mature later. Prefrontal cortex is among the last regions to fully mature, with development continuing into the mid-20s. This protracted development allows extensive experience-dependent refinement, supporting learning and adaptation, but also creates vulnerability to disruption during developmental windows. White matter development continues even longer than gray matter development. Myelination of axons proceeds gradually, with some pathways not fully myelinated until adulthood. Myelination increases conduction velocity and improves the efficiency of information transmission, supporting increasingly sophisticated cognitive functions as development proceeds. The maturation of white matter tracts connecting frontal cortex to other regions parallels the development of executive functions like planning, working memory, and impulse control during adolescence and early adulthood. Sexual dimorphism exists in certain brain structures, though overall brain organization is more similar than different between males and females. Some nuclei in the hypothalamus involved in reproductive function differ in size between sexes. The corpus callosum may show subtle size differences. Certain cognitive functions show average differences between sexes in performance or patterns of brain activation, though with substantial overlap between groups. These differences likely reflect interactions between genetic factors, hormones during development and adulthood, and experience. Aging involves gradual changes in brain anatomy. Brain volume decreases slightly, with greater loss in some regions like prefrontal cortex and hippocampus than in others like visual cortex. White matter integrity declines, particularly in long association pathways. The number of synapses decreases. These structural changes parallel declines in processing speed, working memory, and episodic memory, though other cognitive functions like vocabulary and general knowledge often remain stable or even improve. The brain retains capacity for plasticity throughout life, with learning and experience continuing to modify connections, though the magnitude of plastic changes may diminish with age. Plasticity, the brain's ability to modify its structure and function in response to experience, persists throughout life and operates at multiple levels. At the synaptic level, long-term potentiation and depression strengthen or weaken connections based on patterns of activity. At the cellular level, dendrites can grow or retract, and new synapses can form. At the systems level, cortical representations can expand or contract, and functions can reorganize after injury. This plasticity allows learning, adaptation to changing environments, and some degree of recovery after damage. After brain injury, remaining tissue can sometimes compensate for lost functions. Adjacent areas may assume functions previously performed by damaged regions. Corresponding areas in the opposite hemisphere may contribute. Subcortical structures may support functions typically dependent on cortex. The extent of functional recovery depends on many factors, including the location and size of damage, the age at which injury occurs, and the availability of rehabilitation. Understanding the anatomical basis of plasticity, which structures can reorganize, how connections can be modified, informs strategies for promoting recovery. The anatomical asymmetries between the left and right hemispheres extend beyond language. The right hemisphere generally dominates for spatial attention, face recognition, prosody, the emotional tone of speech, and certain aspects of music perception. The left hemisphere typically dominates for language, mathematical calculation, and fine motor control of the dominant hand. These functional differences reflect underlying anatomical asymmetries. The planum temporal, a region involved in language, is typically larger in the left hemisphere. The right frontal lobe often protrudes further forward than the left, and the left occipital lobe often protrudes further back, creating a subtle torque pattern visible in brain anatomy. Despite these asymmetries, the two hemispheres work together through extensive connections via the corpus callosum and other commissures. Most functions involve both hemispheres to some degree, with each contributing different processing. The popular notion of people being left-brained or right-brained oversimplifies the complex, integrated nature of brain function. Normal cognition and behavior emerge from the coordinated activity of networks distributed across both hemispheres. The integration we observe throughout neuroanatomy, sensory systems converging on association areas, motor systems receiving input from multiple planning regions, emotional and cognitive systems interconnected through prefrontal limbic pathways, hemisphere communicating through commissures, reflects a fundamental principle. The brain does not consist of independent modules performing isolated functions, but rather of interconnected networks that process information in parallel and interact extensively. Perception, action, emotion, and cognition are not separate processes occurring in separate places, but aspects of unified neural activity distributed across networks. Understanding neuroanatomy thus means understanding not just where things are, but how they connect, how information flows through circuits, how activity in one region influences others, how distributed networks create unified functions. The anatomical substrate determines what computations are possible, what information is available, where, how quickly signals can propagate, how systems can interact. Structure and function are inseparable. The anatomy we are mapping is the physical implementation of the processes that create mind and behavior. As we continue toward the completion of our journey, we carry forward this understanding of the nervous system as an integrated whole. Individual structures have specific roles, but these roles emerge from their position within larger networks. Pathways connect structures in specific patterns, determining information flow. Neurotransmitter systems modulate activity across widespread regions. Feedback connections create loops that support learning and homeostatic regulation. And the overall architecture, hierarchical yet highly interconnected, specialized yet integrated, enables the remarkable capabilities of the nervous system. The landscape we have been traversing reveals itself as a territory of extraordinary complexity and elegant organization, where billions of neurons arrange themselves into circuits, circuits into systems, systems into networks that span the entire brain, creating the substrate for every aspect of mental life. This is the achievement of evolution working over vast time, of development unfolding according to genetic programs refined by experience, of learning constantly modifying connections to match behavior to the world. And it is the anatomy that makes you possible, the physical structure that enables consciousness, that transforms sensory input into perception, that generates the actions you take, that stores your memories and shapes your future. We have journeyed through the architecture of the nervous system, from the segmental organization of the spinal cord to the folded expanse of the cerebral cortex, from sensory pathways ascending toward awareness to motor pathways descending toward action, from white matter highways connecting distant territories to the intricate networks underlying memory, emotion, and thought. Now, as our exploration moves toward completion, we examine the remaining systems and draw together the threads that reveal the nervous system as a unified whole. The cranial nerves deserve closer examination, as these 12 pairs extend the reach of the central nervous system throughout the head and neck, and in some cases into the chest and abdomen. Each cranial nerve has a unique anatomy reflecting its specific functions, and together they illustrate the diversity of neural organization. The olfactory nerve, the first cranial nerve, is unusual in that its neurons are not contained within a nerve trunk, but consist of receptor cells distributed across the olfactory epithelium in the nasal cavity. These bipolar neurons have dendrites exposed to the air and axons that pass through tiny openings in the cribriform plate of the ethmoid bone to reach the olfactory bulb. The olfactory system's direct connection to the brain, without a relay through the thalamus, reflects its ancient evolutionary origins and may explain the powerful connection between smell and memory. The optic nerve, despite its name, is not truly a peripheral nerve, but rather a tract of the central nervous system. The axons forming the optic nerve are extensions of retinal ganglion cells, and they are myelinated by oligodendrocytes rather than Schwann cells. The partial crossing of optic nerve fibers at the optic chiasm creates the anatomical basis for binocular vision, with each hemisphere receiving information from both eyes about the opposite visual field. The oculomotor, trochlear, and abducens nerves control the 6 muscles that move each eye. The oculomotor nerve, the third cranial nerve, innervates 4 of these muscles plus the muscle that elevates the eyelid. It also carries parasympathetic fibers that constrict the pupil and adjust the lens for near vision. The trochlear nerve, the 4th cranial nerve, innervates the superior oblique muscle, which rotates the eye downward and laterally. Uniquely, the trochlear nerve emerges from the dorsal surface of the brainstem and is the only cranial nerve to decussate completely. The abducens nerve, the 6th cranial nerve, innervates the lateral rectus muscle, which moves the eye laterally. These 3 nerves originate from nuclei in the midbrain and pons and must coordinate precisely to produce smooth conjugate eye movements. The trigeminal nerve, the 5th and largest cranial nerve, has 3 major branches that provide sensation from the face, scalp, oral cavity, and nasal cavity. The ophthalmic division supplies the forehead, upper eyelid, and anterior scalp. The maxillary division supplies the cheek, upper lip, upper teeth, and palate. The mandibular division supplies the lower face, lower teeth, and anterior tongue, and also carries motor fibers to the muscles of chewing. The trigeminal nerve's 3 divisions converge on the trigeminal ganglion, which contains the sensory neuron cell bodies, then enter the pons as separate roots. Within the brainstem, trigeminal sensory nuclei extend from the midbrain through the medulla, processing different types of sensation: touch, pain, temperature, and proprioception from facial muscles. The facial nerve, the 7th cranial nerve, is primarily motor, controlling the muscles of facial expression that create the subtle movements conveying emotion. It also carries parasympathetic fibers to lacrimal glands and salivary glands, and taste fibers from the anterior 2/3 of the tongue. The facial nerve emerges from the brainstem at the junction of pons and medulla, travels through the internal acoustic meatus along with the vestibulocochlear nerve, then takes a winding course through the temporal bone before emerging to distribute across the face. Damage to the facial nerve produces characteristic paralysis of facial muscles, inability to close the eye, and loss of taste on the affected side. The vestibulocochlear nerve, the 8th cranial nerve, consists of 2 functional components sharing a common path. The cochlear division carries auditory information from the cochlea to cochlear nuclei in the medulla. The vestibular division carries information about head position and movement from the semicircular canals and otolith organs to vestibular nuclei in the medulla and pons. These 2 sensory systems, though anatomically linked, serve different functions. One creates our perception of sound, the other our sense of balance and spatial orientation. The glossopharyngeal nerve, the 9th cranial nerve, has diverse functions reflecting its multiple components. It carries sensory information from the posterior tongue and pharynx, taste from the posterior third of the tongue, and visceral sensory input from carotid body chemoreceptors and baroreceptors that monitor blood oxygen and pressure. It provides motor innervation to pharyngeal muscles involved in swallowing and carries parasympathetic fibers to the parotid salivary gland. This multiplicity of functions in a single nerve reflects the complex innervation required for the pharyngeal region. The vagus nerve, the 10th cranial nerve, has the most extensive distribution of any cranial nerve, sending branches throughout the neck, thorax, and abdomen. Its name derives from the Latin for wandering, reflecting its far-reaching course. The vagus carries sensory information from the external ear, pharynx, and larynx, visceral sensation from thoracic and abdominal organs, and taste from the epiglottis. It provides motor innervation to muscles of the pharynx and larynx involved in swallowing and speech. Most importantly, it carries extensive parasympathetic innervation to the heart, lungs, and digestive organs throughout the thorax and most of the abdomen. Through these widespread projections, the vagus nerve influences heart rate, respiratory function, digestive motility, and many other visceral processes. The accessory nerve, the 11th cranial nerve, has an unusual anatomy. It consists of a cranial part that joins the vagus nerve and a spinal part that originates from cervical spinal cord segments, ascends through the foramen magnum into the skull, then exits again to innervate the sternocleidomastoid and trapezius muscles of the neck and shoulder. This circuitous route reflects the nerve's developmental origins and evolutionary history. The hypoglossal nerve, the 12th cranial nerve, innervates the intrinsic and most extrinsic muscles of the tongue, controlling the tongue movements essential for speech and swallowing. The nerve emerges from the medulla, exits the skull through the hypoglossal canal, and descends in the neck before distributing to the tongue muscles. Damage to the hypoglossal nerve causes the tongue to deviate toward the affected side when protruded, as the unopposed muscle on the healthy side pushes the tongue across the midline. The diversity of cranial nerve anatomy, some purely sensory, others purely motor, many mixed, some with parasympathetic components, some confined to the head, others extending into the body, reflects the complex innervation requirements of the head and neck region and the special senses. Understanding cranial nerve anatomy is essential for clinical neurology, as lesions affecting specific nerves produce characteristic patterns of deficits that help localize damage. The autonomic nervous system's anatomy warrants deeper consideration, as this system regulates virtually every organ and maintains homeostasis through continuous, unconscious adjustments. Beyond the basic division into sympathetic and parasympathetic components, the autonomic system shows sophisticated organization that allows precise, organ-specific control. The sympathetic chain ganglia, running alongside the vertebral column from cervical to sacral levels, provide distributed sites where preganglionic fibers synapse onto postganglionic neurons. Not all preganglionic fibers synapse at the same level they exit the cord. Some ascend or descend through the chain before synapsing, allowing sympathetic outflow from thoracic and lumbar segments to reach cervical and sacral targets. Some preganglionic fibers pass through the chain without synapsing and continue to collateral ganglia, closer to target organs. The celiac, superior mesenteric, and inferior mesenteric ganglia that supply abdominal organs. This anatomical arrangement allows for both widespread, coordinated sympathetic activation and more localized responses. During stress or exercise, broad activation of the sympathetic system produces systemic changes. But sympathetic activity can also be locally regulated. For instance, blood vessels in different organs can be independently controlled to redirect blood flow to where it is most needed. The parasympathetic system's anatomy, with preganglionic fibers extending all the way to or near target organs before synapsing, allows more discrete control. Parasympathetic ganglia are typically small and located on or within the organs they innervate. This means postganglionic fibers are very short, and parasympathetic effects tend to be more localized than sympathetic effects. The vagus nerve's extensive distribution allows parasympathetic influence over a wide range of organs. But each vagal branch can be independently regulated, providing flexibility. A third division of the autonomic system, the enteric nervous system, consists of neural networks embedded in the walls of the gastrointestinal tract. These networks contain hundreds of millions of neurons organized into two main plexuses: the myenteric plexus between muscle layers and the submucosal plexus in the submucosa. The enteric nervous system can operate independently of central control, generating the rhythmic contractions that move contents through the digestive tract and regulating secretion. However, it receives input from sympathetic and parasympathetic systems that modulate its activity, and it sends sensory information back to the central nervous system through vagal and spinal afferents. The complexity of the enteric nervous system, its large number of neurons, diverse cell types, intricate circuits, and partial independence from central control, has led to it being called the second brain. Its anatomy allows the digestive system to function with considerable autonomy while remaining coordinated with overall physiological state through autonomic connections. Neurotransmitter systems form another layer of organization, with specific populations of neurons using specific chemical messengers and projecting to widespread targets. These systems modulate neural activity across large regions, influencing arousal, attention, mood, and many other functions. The cholinergic system includes motor neurons that release acetylcholine at neuromuscular junctions, preganglionic autonomic neurons, postganglionic parasympathetic neurons, and several populations of central neurons. In the brain, cholinergic neurons in the basal forebrain project widely to cortex and hippocampus, promoting arousal and attention and supporting learning and memory. Cholinergic neurons in the brainstem contribute to arousal and rapid eye movement generation. Loss of basal forebrain cholinergic neurons is associated with Alzheimer's disease, and cholinergic dysfunction contributes to the memory impairment characteristic of this condition. The noradrenergic system originates primarily from the locus coeruleus, a small nucleus in the pons. Despite containing only about 15,000 neurons per side in humans, the locus coeruleus sends projections throughout the brain and spinal cord. Noradrenergic projections influence arousal, attention, stress responses, and many other functions. The locus coeruleus activates in response to novel or salient stimuli, facilitating orienting and behavioral responses. The serotonergic system originates from the ref nuclei, a series of midline nuclei extending through the brainstem. Like the noradrenergic system, serotonergic projections reach widely throughout the brain and spinal cord. Serotonin influences mood, anxiety, impulse control, appetite, and other functions. Many antidepressant medications act by increasing serotonin availability, highlighting the importance of this system in emotional regulation. The dopaminergic system includes several pathways with different origins and targets. The nigrostriatal pathway, originating in the substantia nigra and projecting to the striatum, is crucial for motor control and degenerates in Parkinson's disease. The mesolimbic pathway, originating in the ventral tegmental area and projecting to the nucleus accumbens and other limbic structures, processes reward and motivation. The mesocortical pathway, also from the ventral tegmental area, projects to prefrontal cortex and contributes to cognitive function. The tuberoinfundibular pathway from the hypothalamus to the pituitary, regulates hormone secretion. These anatomically distinct dopamine pathways subserve different functions, and dysfunction in different pathways contributes to different disorders. Other neurotransmitter systems include histaminergic neurons in the hypothalamus that promote wakefulness, orexinergic neurons in the lateral hypothalamus that stabilize arousal states, and various populations using neuropeptides to modulate neural activity. These diffuse modulatory systems act in concert with point-to-point connections to shape neural processing across the brain. The blood supply to the brain and spinal cord deserves detailed consideration, as vascular anatomy directly impacts clinical outcomes in stroke and other vascular disorders. The internal carotid arteries supply anterior circulation, giving rise to the anterior and middle cerebral arteries. The vertebral arteries merge to form the basilar artery, which supplies the brainstem and cerebellum and gives rise to the posterior cerebral arteries. The circle of Willis, formed by communicating arteries connecting these major vessels, provides collateral circulation. Though the configuration varies considerably between individuals and is incomplete in many people, penetrating arteries arise from major vessels and dive into brain tissue, supplying deep structures. The lenticulostriate arteries, branching from the middle cerebral artery, supply the basal ganglia and internal capsule. These small arteries are prone to hemorrhage and hypertension, producing deep brain bleeds. The thalamoperforating arteries supply the thalamus. Occlusion of these small vessels can produce pure sensory strokes affecting the thalamus. Venous drainage follows a different pattern from arterial supply. Superficial veins on the cortical surface drain into dural venous sinuses, large channels within the dura mater. The superior sagittal sinus runs along the top of the brain in the midline, collecting blood from superficial cortical veins. The transverse and sigmoid sinuses carry blood laterally and inferiorly to the jugular veins. The straight sinus drains deep structures. Unlike arteries, cerebral veins have no valves, allowing bidirectional flow under certain conditions and potentially enabling spread of infection from facial veins into intracranial venous sinuses. The spinal cord receives blood from a single anterior spinal artery and paired posterior spinal arteries, supplemented by radicular arteries entering at intervals. The anterior spinal artery supplies the anterior two-thirds of the cord, including motor pathways. The posterior spinal arteries supply the posterior third, including sensory pathways. This vascular anatomy explains why anterior spinal artery occlusion produces motor deficits and loss of pain and temperature sensation, while preserving fine touch and proprioception carried in the dorsal columns supplied by posterior spinal arteries. Throughout our exploration, we have encountered the principle of localization, that specific brain regions support specific functions. Yet we have also seen that most functions involve distributed networks rather than single regions. This apparent paradox resolves when we recognize that regions are specialized for particular types of processing, but complex functions require multiple types of processing occurring in coordinated fashion across multiple regions. Vision illustrates this principle beautifully. The primary visual cortex extracts basic features like edges and orientations from the retinal image. Surrounding visual areas process progressively more complex features: color, motion, shapes, objects. The ventral stream through temporal cortex specializes in identifying objects, while the dorsal stream through parietal cortex specializes in locating them and guiding actions toward them. Face recognition involves fusiform regions. Place recognition involves parahippocampal regions, and biological motion perception involves the superior temporal sulcus. Yet visual perception emerges from the integrated activity of this distributed network, with information flowing forward from primary areas, backward through feedback connections, and laterally through association fibers. Similarly, motor control involves primary motor cortex generating commands, premotor and supplementary motor areas planning sequences, parietal regions providing sensory guidance, basal ganglia selecting actions and controlling initiation, cerebellum refining movements, and brainstem and spinal circuits executing patterns. Damage to any component impairs movement, but in different ways, reflecting that component's contribution to the overall process. Language involves auditory cortex for speech sounds, visual cortex for written words, Wernicke's area for comprehension, Broca's area for production, motor cortex for articulation, and distributed semantic networks for meaning. Memory formation involves hippocampus binding elements into coherent episodes, but retrieval of consolidated memories depends on distributed cortical networks, where different aspects of experience are stored in the regions that originally processed them. This distributed yet specialized organization reflects the brain's evolutionary history and developmental constraints. Brains evolved through modification of existing structures rather than redesign from scratch, resulting in circuits that work but that might be organized differently if designed de novo. The cortex expanded dramatically in mammals, particularly primates, but maintained its basic six-layered structure and columnar organization. New areas emerged through expansion and differentiation of existing cortex, creating the mosaic of specialized regions we observe. The folding of the cortex into gyri and sulci while increasing surface area is not random, but follows consistent patterns that reflect underlying organization. Primary sensory and motor areas tend to occupy particular gyri. Association areas fill the space between these primary regions. The patterns of folding, while similar across individuals, show enough variation that precise boundaries between functional areas are not always aligned with anatomical landmarks. This variation underscores that function depends on underlying circuitry and connections, rather than solely on gross anatomy. The development of imaging technologies has revolutionized our ability to study brain anatomy in living humans. Structural imaging with magnetic resonance reveals detailed anatomy, showing gray and white matter distribution, identifying structures, detecting abnormalities. Diffusion imaging traces white matter pathways by measuring water diffusion along axons, allowing reconstruction of major fiber bundles in individual brains. Functional imaging reveals which regions activate during different tasks, showing the neural correlates of cognitive processes. These techniques complement traditional anatomical methods based on postmortem examination and allow correlation of structure with function in ways not previously possible. Imaging studies have revealed that the brain's anatomy is not fixed in adulthood, but continues to change with experience. London taxi drivers, who must learn detailed spatial knowledge of the city, show enlargement of posterior hippocampus, a region involved in spatial memory. Musicians show structural differences in auditory and motor regions reflecting their extensive practice. Bilingual individuals show differences in language-related areas. These findings demonstrate that anatomy and function are bidirectionally related. Anatomy constrains what functions are possible, but experience and behavior shape anatomy. The concept of the connectum, a comprehensive map of neural connections in the brain, represents an ambitious goal to chart the complete wiring diagram of the nervous system. At the microscopic level, this means identifying every neuron and every synapse, a goal achieved so far only for the nematode C, elegans with its 302 neurons. For larger brains, including humans, this level of detail remains beyond current technology. At the macroscopic level, mapping major pathways and connectivity patterns between regions is feasible and increasingly being accomplished. These efforts reveal network properties, which regions are highly connected hubs, which pathways carry the most traffic, how information can flow through multiple routes. Network analysis reveals that brain connectivity follows small world architecture, with most regions connected to nearby neighbors, but also connected through a few long-range pathways to distant regions. This organization supports both local processing and global integration while minimizing wiring length. Certain regions, particularly in association cortex, serve as hubs with connections to many other regions, allowing them to integrate information from diverse sources. Damage to these hub regions tends to produce more widespread deficits than damage to more locally connected regions. The brain also exhibits modular organization with communities of regions that are more densely interconnected with each other than with regions outside the community. These modules often correspond to functional systems. Visual regions form a module, motor regions another, language areas another. Yet modules are not isolated but communicate through intermodular connections that allow different functional systems to interact. This modular yet integrated architecture supports both specialized processing within systems and coordination between systems. As we approach the completion of this anatomical journey, we return to fundamental questions about the relationship between brain structure and mental function. How does the physical anatomy we have mapped give rise to consciousness, to the subjective experience of perceiving, thinking, and feeling? This question, sometimes called the hard problem of consciousness, remains unsolved. We can identify neural correlates of consciousness, brain activity patterns associated with conscious awareness, and we know that certain structures, particularly the thalamus and cortex, are necessary for consciousness. But how and why neural activity creates subjective experience remains mysterious. What we can say is that the anatomical organization we have explored provides the substrate necessary for consciousness. The extensive interconnectivity of cortical regions, the integration of information from multiple sensory modalities, the feedback loops between cortex and thalamus, the ability to maintain representations in working memory, the mechanisms for attention that select relevant information. All these anatomical features contribute to creating unified, coherent experience from the fragmentary input reaching our senses. The human brain's anatomy reflects both our shared evolutionary heritage with other animals and the unique features that distinguish human cognition. The basic organization, spinal cord, brainstem, cerebellum, diencephalon, cerebrum, is shared with other mammals. The fundamental circuitry of sensory and motor systems, the organization of the autonomic system, the structure of limbic regions, these are variations on themes found throughout vertebrates. But humans show disproportionate expansion of association cortex, particularly in prefrontal and temporal regions. We show increased connectivity between cortical areas. We show greater asymmetry between hemispheres. These anatomical differences correlate with human cognitive capabilities, language, abstract reasoning, complex planning, sophisticated social cognition. Yet individual human brains vary considerably in their precise anatomy. Brain size varies across a twofold range in normal individuals. The patterns of gyri and sulci, while generally consistent, show individual variation. The exact sizes and locations of functional areas differ between people. These variations likely contribute to individual differences in cognitive abilities, personality, and susceptibility to neurological and psychiatric disorders. Understanding normal variation is as important as understanding average anatomy. The anatomy we have explored represents the healthy adult nervous system. But this anatomy can be disrupted by injury, disease, or developmental disorders, producing the symptoms that bring people to clinical attention. Stroke damages tissue in vascular territories. Tumors displace and destroy structures. Degenerative diseases like Alzheimer's, Parkinson's, and amyotrophic lateral sclerosis selectively affect particular populations of neurons. Developmental disorders like autism and schizophrenia may involve subtle alterations in brain structure and connectivity. Trauma can cause focal damage or diffuse axonal injury. Understanding normal anatomy provides the foundation for understanding how these pathologies produce their symptoms and for developing interventions to prevent or treat them. The nervous system's capacity for plasticity and compensation means that anatomical damage does not always produce permanent deficits. The brain can reorganize, with remaining tissue assuming functions previously performed by damaged regions. The extent of recovery depends on many factors: the location and size of damage, the age at which it occurs, the availability of rehabilitation. Understanding the anatomical basis of plasticity, how connections can be modified, how functions can reorganize, informs rehabilitation strategies and offers hope for recovery after injury. As this exploration draws toward its close, we find ourselves returned to where we began, observing the nervous system as an integrated whole, a vast network of interconnected structures that process information, generate behavior, and create the mind. We have traced pathways carrying sensation and directing movement. We have examined regions processing vision and sound, storing memories and regulating emotions, planning actions and producing speech. We have observed the white matter highways connecting distant territories, the chemical messengers modulating widespread activity, the protective structures shielding delicate tissue, the blood vessels supplying metabolic needs. Throughout this journey, certain themes have recurred. Topographic organization creates orderly maps of sensory surfaces and motor outputs. Hierarchical processing extracts increasingly complex features through successive stages. Parallel pathways process different types of information simultaneously. Feedback connections allow higher levels to influence lower ones. Specialized regions contribute particular types of processing, but complex functions emerge from the coordinated activity of distributed networks. And the whole system exhibits remarkable plasticity, continuously modifying connections based on experience while maintaining the stable patterns that constitute our identity. The nervous system you possess, whose anatomy we have been exploring, is the most complex structure in the known universe. It contains approximately 86 billion neurons, each connected to thousands of others through hundreds of trillions of synapses. The number of possible patterns of activity across this network exceeds the number of atoms in the observable universe. This vast interconnected structure operates continuously, in parallel at multiple scales, molecules diffusing across synapses, electrical signals racing along axons, populations of neurons oscillating in synchrony, networks reorganizing based on experience. And yet, for all its complexity, the nervous system follows comprehensible principles. Its anatomy reflects solutions to functional challenges: how to process sensory information efficiently, how to coordinate movement precisely, how to store and retrieve memories, how to regulate internal states, how to adapt to changing circumstances. Understanding the anatomy means understanding the physical implementation of these solutions, seeing how evolution and development have shaped tissue into circuits that support life and mind. As you rest in this moment, that tissue continues its work. Sensory neurons throughout your body send streams of signals toward the central nervous system. Ascending pathways carry this information through the spinal cord and brainstem to the thalamus and cortex. Processing occurs through hierarchies of regions, extracting meaning from raw sensation, creating the rich perceptual world you experience. Descending pathways maintain muscle tone, regulate breathing, adjust heart rate, coordinate the countless automatic processes that sustain you. Networks in the medial temporal lobe consolidate the information you have encountered, creating memories that may persist for a lifetime. Modulatory systems set the overall tone of neural activity, maintaining the state of quiet wakefulness. And the vast association networks of the cortex integrate all of this into unified, coherent experience. The sense of being present, of existing as a conscious entity embedded in the world. The anatomical structures we have explored are not abstractions or distant objects of study, but the very tissue that makes your experience possible. The visual cortex that processes what you see, the auditory cortex that interprets sounds, the motor cortex that directs your movements, the prefrontal cortex that formulates your thoughts, the hippocampus that creates your memories, the amygdala that colors experience with emotion. All of these are present, active, functioning within you right now as you exist in this moment. This knowledge connects you to something larger, to the evolutionary history that shaped these structures over millions of years, to the developmental processes that constructed your nervous system, to the shared anatomy that you hold in common with every other human and that links you to all other living things through the deep similarities of nervous systems across species. The anatomy we have mapped is simultaneously uniquely yours and universally shared, shaped by your individual experience, yet following patterns found in all brains. There is a quiet wonder in this understanding, a recognition that the physical structures we can identify, name, and trace through the landscape of the nervous system give rise to everything you are and everything you experience. The self is not separate from the brain, but emerges from it, from the patterns of connectivity, the flow of information through circuits, the modulation of activity across networks. Understanding neuroanatomy is thus a form of self-knowledge, an appreciation of the substrate of consciousness. As our journey completes its circle, returning to the starting point enriched by all we have traversed, rest in the awareness that these systems continue their ancient work. The pathways we traced remain open, carrying signals. The structures we examined remain active, processing information. The networks we explored maintain their configurations, holding in their patterns of connection the accumulated experience of your life. All of this proceeds without requiring conscious oversight, guided by mechanisms refined through evolution and development, operating with a reliability that allows you to simply exist, to experience, to be. The neuroanatomy we have explored together forms the foundation of who you are, the physical landscape where thought arises, where perception emerges, where memory resides, where intention forms. It is a landscape of extraordinary beauty and complexity, of elegant organization and adaptive flexibility, of structure that enables function and function that shapes structure. And it is a landscape that belongs to you, that is you, continuing its work in every moment, sustaining the remarkable phenomenon of conscious life. The exploration is complete. The map has been drawn. The territory remains alive and active, the architecture of mind made manifest in tissue, the anatomy of awareness itself.